they have this survey that they want you guys to do. It used to be on paper, but obviously there's a lot of us in here, so we're trying to reduce that paper amount. So if you look, and these are on every single table. So what I'm going to ask of you guys, I realize that you're worried about your finals. This is the last thing you want to kind of deal with. But on your way out, if you guys could either scan this with your phone or go to the link that they have here. It's a very short survey on your experience with this session. The feedback that we get from that helps fund studying, okay? So, you know, if you like the fact that they're able to put on all these sessions for finals, filling out the survey is going to help this keep going. And you don't have to just say positive things or whatever. You can say however you feel. But anyway, please do that on your way out. I'd really appreciate it. They'd really appreciate it. Something else that I want to mention is that even though we're having the review today, we do have 2 more SI sessions, okay? They're both tomorrow. It's the same times you've always had, but I'll remind you, we have 11:30, okay? And this is in TA 322, okay? The other one that we have is at 1:30. It's in Classroom Building 105. If you are interested in coming to either of these, you're more than welcome to. But again, just do remember that that will follow our normal SI model. So if you've never been to SI before, what we do here is a little bit different than what we do there. So just kind of letting you know ahead of time. But you're more than welcome to come. I'd be really happy to see you guys. And that's going to be it for us for the semester. So we'll go ahead and get started. The quality isn't the best here, and I apologize for that. But it's kind of the best that we can work with, and hopefully you guys have a copy of this. It is posted on the Google site, so if you want to follow along there or on paper, that might be a little bit easier for you. But we're going to go ahead and launch into this, and you know, just like in any normal review, do raise your hand and let me know if you have any specific questions. I was kind of joking about it earlier, but I'm not the tallest person, and so the way that this computer is positioned, there's a good portion of you guys that are kind of out of my vision range unless I move. So if you want to get my attention, like, please do so. Please do interrupt me. This is for you, okay? The other thing, the last thing that I do want to mention is that this is being filmed. You will not be on film, don't worry about that. But if you want to watch this later, you can do that. So you have that option. That's with a couple of sessions here. All right, any questions before we get started? We're not going to have our break in the middle like we usually do. Instead, we're going to power through as hard as we possibly can. And then we're going to try and save time at the end for any other questions that you guys want to cover. My review is longer than your final. Your final is 30 questions, and I hope that this review is a little bit more challenging. The reason that I do this is because if you feel good about this, you should really feel good about that. You know what I mean? So the other thing too, I realize I have a lot of caveats here. I apologize for this. The other thing as well is, you know, just like any other time, this is not comprehensive. This is an entire semester's worth of what you've learned in like 40-something questions. So there's no way that it's going to cover everything, so please don't disregard stuff that I don't have on here. My idea here is just to kind of get you to start remembering these things and give you a platform for your studying. That's why I did it so early. Okay? Let's go ahead and get started. Okay. So our first one, we're going back to chapter 1. These kind of follow a chapter progression. I'll kind of let you know when we break to different chapters. Species that are in the same blank are more closely related than species that are only in the same blank. So we're getting back to what we had talked about in the very beginning with taxonomy, right? So I'm going to write on the side here, we have, okay, so we have kingdom, phylum, class, order, family, genus, and species. Hopefully you guys can see that. I think it's a little bit clearer on the back. So what this means, again, you can't just remember the order, but it is important to remember the order. You have to understand what this means. So just drawing back to what we talked about for the first exam, as we go down, we become more specific. Okay? Let me give you an example. We are of the genus and species Homo sapiens, right? So that's this, down at the bottom. However, we're of the kingdom Animalia, right? So we are animals, so are snails, so are chickens, so are giraffes. You know what I mean? So there's a lot of animals, right? But we are one species. So the further down you go, the more specific it is. Another way to say it is the more closely related it is. That's the connection here, okay? So if something is more closely related, what this, brass tacks is, what this means for us is that it's lower on the list, okay? So we're looking basically for our first item to be lower than our second item, okay? So let's analyze our choices. We have phylum and class. Well, that's not going in the right order that we want. We have family and then order. That seems to be going in that path. I'm going to flag it. Just in case our logic is flawed here, we're going to analyze all of our choices. We have family and then genus. We have kingdom and then phylum. And then we have class and order. So our answer is B. Questions? Again, I'm going to mention here our standard test-taking strategy of if you don't know what's going on, like say you forget how to analyze this, but you remember the order, if you compare them, there's only one choice here that is not like the others. So we're going to kind of take this back to Sesame Street rules here. Pick the odd one out, and that tends to be the answer. So, there's a lot of ways to work around maybe not remembering all the fine details. But again, we do want to kind of try and get your memory back up to speed. The next one, we're going to do some chemistry. What is the maximum number of covalent bonds that oxygen can make with hydrogen? And also notice how I'm underlining lots of things. I'm getting engaged with it. I'm not just doing this for your benefit. This is exactly everything that I'm writing on here, for the most part, is what I would write if I was taking this exam myself. The fact that you're able to write on this exam is not something that you should take for granted. You should really exploit that and use it to your advantage. Okay, so we're looking at covalent bonds. I'm just going to say, what do I remember about this? We're sharing. We're going to talk about that in a question a little bit later. We're talking about oxygen. We're talking about hydrogen. What's the rule that Professor Thomas taught us? Honk, right? Cool. So HONC, again, another thing where it's one thing to have memorized this. It's another thing to be able to apply it. That's the level we need to be at. So we have honk, 1, 2, 3, 4. What this means is not how many valence electrons they have. Well, actually that technically is the case with carbon. But it does not necessarily mean that. What this necessarily means for you is this is how many bonds each of them would want to make. Like the maximum number of bonds it can make without breaking rules and getting into some other stuff. Okay? So we're talking about oxygen, okay? And it wants to make 2 bonds, right? And we have hydrogen, and it wants to make 1 bond. Is hydrogen going to be able to make 2 bonds just because oxygen wants to? No. So what does our answer have to be? 1. Our answer is 1. We could put 2 hydrogens on here, but what we're talking about is 1 oxygen and 1 hydrogen atom. How many bonds can they make together? They can only make 1. You're sort of reduced down to your limiting element here, which is going to be hydrogen. Does that make sense to everyone? What's happening here? Because just because oxygen is able to do more doesn't mean that hydrogen is capable of that. Okay? Alright. If you guys end up thinking of questions and want me to backtrack, I absolutely can as well. Okay, so now we have nitrogen And you're like, okay, well, we didn't learn that much about nitrogen. This is coming back to our first exam review, but I really like this question because it's very indicative of the types of questions that are offered in this class, where the specific example that we're working with, maybe you're not that familiar with, but the rules you are, and so you have to apply those rules to your new situation. So we have nitrogen. Let's say we know absolutely nothing about it. That's fine. You don't have to. But it's much more electronegative. That's a term that we've heard before than hydrogen. Okay? So nitrogen is more electronegative. That's something that's very important here. So then we need to figure out what's correct about the atoms in ammonia. So I'm going to draw ammonia. And again, maybe you don't know how to draw it, and that's okay. But if you remember that hydrogen can only make one bond, you kind of only have one option here. And then nitrogen can make three. So we actually are following those rules, right? But let's go ahead and analyze this. So we said nitrogen is really electronegative. That means it's an electron hog. It wants all of the electrons. It actually has an extra lone pair here. But it's going to take on this negative charge. Okay? So if it's pulling all of the negative charge, what is going to be the relative charge of the hydrogen? It's going to be positive, right? If one thing is more negative, everything is like a yin and yang. Just like when we talked about acid-base, nothing happens in a vacuum. Everything is give and take. So if one thing goes up, another thing goes down. So if this takes all the negative, then these guys get the positive. Okay? I have a very important question for you that's not actually directly addressing these choices. What type of molecule do we call this? Your options, you have like ionic, you have covalent, you have polar covalent. How do we feel? It is polar and it is covalent. So that's, you should be able to analyze these things. If something is polar, that means that there's more charge on one side than the other in that molecule or in that bond, depending on what you're looking at. Okay, so now let's look at our choices. The nitrogen atom has a partial positive charge. What do we think? No. Okay, talk to me, guys. I need to know that you are alive in this room. I know it's a Sunday. The nitrogen atom has a strong positive charge. What do you think about that? No. Thank you. There are covalent bonds between the hydrogen atoms. No, because that would be like bonds in between these, and there's no way, because that would mean that a hydrogen was taking on two bonds. The only way for that to work would be if a hydrogen had two bonds. Each hydrogen atom has a partial positive charge. You think so? What about each hydrogen atom has a slight negative charge? No. So see, once we draw our picture, everything kind of plays itself out pretty nicely. Questions here? Okay. What is the difference between a covalent and ionic bond? So we're already starting to talk about this, okay? But it boils down to something very simple. It's what we do with the electrons. Electrons are what we focus on because if you change the protons, what do? You change the element. Yeah, exactly. So so you can't change the number of protons, therefore we're going to have to look at the electrons. So covalent bonds share electrons. That's like the most simple way to put it. But ionic bonds involve like a swap of electrons or a trading of electrons, something to that effect. So one element gets electrons, one element loses electrons. So that is all we need to answer this question. Okay? So covalent bonds involve the sharing of neutrons. How do we feel about that? Did we ever talk about sharing neutrons, ever? No. We're focused on electrons, okay? Covalent bonds involve the sharing of protons. Also no. You see how we don't even have to read the whole answer choice to do this? But there's a lot of words, and it's intimidating. But you have to just tackle it one piece at a time, and then it's not a problem. Covalent bonds involve the transfer of electrons between atoms. Does that sound right? No, that sounds a lot more like ionic, right? And then ionic bonds involve the sharing of neutrons. So yeah, we have a lot of issues there. So that's wrong. Okay, covalent bonds involve the sharing of protons. That is also wrong, and so we actually end up here. Covalent bonds involve the sharing of electrons. That's good. And ionic bonds involve the electrical attraction between atoms. So we didn't actually mention that, but this is correct. So what happens is, for instance, if we end up with, let's say we have sodium and we have chlorine. This is like the classic example of ionic bonding, okay? Chlorine has 7 valence electrons. Sodium has 1. Do you think it's easier for sodium to take 7 or give away 1? Give away 1. And then chlorine wants 1, so it's like a match made in heaven. So sodium's going to give its 1 to chlorine. Okay? And then what kind of charge do electrons have? Negative. So if you give away a negative, what are you left with? Positive. So sodium becomes a positive, and then chlorine that took on that negative is now going to have a negative 1 charge. And so then what happens is they're like magnets. They're going to want to stick together because positive and negative are attracted to each other. So that's what that's saying. Okay? Good? All right. Okay. Which of the following molecules orient themselves into sandwich-like membranes because of hydrophobic components within the membrane? That's quite a mouthful. We have a sandwich-like thing, so maybe there's like a layer because of hydrophobic components within the membrane. What does this sound like to you? It sounds like phospholipids, right? And we can actually draw them briefly to remember what these look like. So, these are a couple of phospholipids. Let me draw the other side of our layer here. So we have these polar regions. And then we have this nonpolar. Also, remember that polar and hydrophilic are synonymous, and nonpolar and hydrophobic are synonymous. But yeah, so, really these terms can be used interchangeably. I'm just going polar, nonpolar, because it's a little bit easier to say. So, again, it's saying because of hydrophobic components, what's on either side of this membrane that the tails might not like? We're talking about a cell here. Water. Cells have water, right? Water's polar. So it's kind of like if you have salad dressing in your fridge. Let's say you have some generic Italian dressing in a bottle, and you pull out the bottle, and there's all of the seasonings and everything on the bottom, and then you have this huge glob of oil on the top, right? The oil is trying to hide from the water, or it's trying to avoid it as much as possible because it does not want to be involved. That's kind of what's happening here with these tails. It's like, well, we got to make the best of a bad situation. Let's just huddle in the center and avoid the stuff that we don't like, which is the polar water. Okay? Trailblazing. Okay. Hydrophilic properties are characteristic of all of the following except. So what did we just say hydrophilic was? Polar, right? So we're going to go ahead and eliminate this choice. Again, remember we're saying except here. Are polar things soluble in water? Yeah, they are. Because like goes with like, right? Molecules that readily ionize in water. Maybe we're not sure. I'm going to flag this. Okay? The long hydrocarbon chain components of some molecules. Do we know? Maybe? Yes, no? Well, hydrocarbons means we have lots of C's and we have H's. And this is just like what the tails of your phospholipids look like. It's these fatty acid tails. So that is actually looking like a culprit here. Then we have the hydroxyl group, which is what again? OH? OH is definitely polar because the oxygen is going to pull those electrons. So our answer here is going to be D. Okay? Because when we talk about long hydrocarbon chains, that's pretty much textbook definition of a fat or a lipid, which are hydrophobic. Okay? Huh? What about C? Well, molecules that ionize in water tend to be hydrophilic because ionizing and being soluble are sometimes interchangeable. Yeah, so that's what that boils down to. But I'm just kind of like playing the tone of like, you know, let's say you're not really sure what's going on. So we're going to go with minimum information here. Okay, cool. Let's keep going. We're going to do some of your favorite, which is pH. Yay. Okay. If the H+, remember how to interchange between these. And make sure if you get pH questions that you're reading it very carefully. Really with any question here. There's so many times where I've heard people looking at their exams after, and you guys knew the information, but you read it as something different. So just please take your time. She's giving you literally as long as you want for this 30 question test. So do take your time with it and read it very carefully. So we have the H+ concentration, okay, is 1 times 10 to the negative fifth, okay? So I'm going to just rewrite this here. H+, the little brackets mean concentration, okay? 1 times 10 to the negative fifth, or you could just say 10 to the negative fifth if you don't want to include that 1. Okay? What is the POH? We have a couple different avenues that we can follow to try and figure this out. Okay? So here's option 1. Option 1 says, let's find the pH first. What's the pH of this? Should be 5. Yeah, should be 5. So then, from that, we know that the pH plus the POH has to equal what? 14. So what is the POH? 9? Everyone's got to think about that. 9 plus 5, yes. Yeah, so the POH is 9, okay? Let's try a different method. Let's say you want to do this differently. So we have, so I'm going to put option 2 here. H plus is this. Let's say we want to do OH minus. When we multiply the H plus with the OH minus, they equal 10 to the negative 14. Okay? And when you multiply numbers with exponents, you have to add the exponents, right? With the same base number. So what we're going to say here is that the OH is going to be 1 times 10 to the negative 9. And then from there, you pull the POH, which is 9. Either way will get you there. Really, I guess there's also kind of an option C, which involves you just looking at that and doing one of these in your head. But I'm drawing this out to show the thought process. Everyone good with this? Throwing my pen. Okay. How can peptide bonds within a protein be broken? Okay. So, I want you guys to think about this for a second. What do we think it is? Hydrolysis? All of the above? Interesting. Well, all of the above includes once formed peptide bonds cannot be broken. So, I'm going to eliminate that, and I'm going to eliminate D. Well, give me a moment, and we'll see, okay? If we're, although I do agree, I should have Yeah, okay. So you know what? Let's go under the presumption that all of the above does mean everything above it, because I should have said, like, all of these choices are correct, but I did not, so I apologize. However, all of them are not correct. Okay? When we talk about a protein is a polypeptide, right? A polypeptide is what? In general. That's like more specific. What chapter is this from? What's the title of chapter 5? Macromolecules. Proteins are macromolecules, right? Okay, how do we put together a macromolecule? Well, peptide bonds are used to put together specifically amino acids to make a polypeptide, but we use a specific method. We use dehydration, synthesis, builds things. It takes out a water molecule and then puts two monomers together. Remember this? So then we have hydrolysis, and we do, this analysis here, hydrolysis, okay? So we add water to split. The other choice that we have here, which is denaturation, that involves if we have different levels of protein structure built up, and then something happens, right? The temperature is wrong, the pH is wrong, the salt content is wrong, and we lose all of those hydrogen bonds from like the secondary, tertiary, quaternary structure. But that primary structure can only be broken with hydrolysis. Yeah, so I mean, the biggest thing to keep in mind here with denaturation, that's actually the chemical reaction that occurs when you cook an egg is denaturation of those proteins in the egg. So next time you're having scrambled eggs, you can think about that. But denaturation is involved with breaking the weaker bonds that form the overall shape of a protein. Right? And then we just strip it down and turn it back into that string that we started with, right? But we're talking about breaking peptide bonds. Peptide bonds are part of that core string. The only way to break them is through hydrolysis. Okay. Good here? All right. The cellular structure that is involved in producing ATP during aerobic respiration is... Anybody? Mitochondria. You say yes, but you didn't tell me what you thought it was. There's too much pressure in this room, I think. We're all friends here. It's okay. You can tell me what you think. Is anyone 100% ready to walk into the exam, like right after this? Yeah, okay. So it's fine. We're all friends here, right? We can get things wrong. It's okay. Which of the following cellular structures are common to both prokaryotes and eukaryotes? Yeah, I'm sorry. Ribosomes? Tends to be a popular choice. Well, let's think about this. So, what do prokaryotes not get to have? They get no membrane bound organelles. Right? Remember that? So what's off the table here? Nucleoli. That's a nucleolus, which is part of a nucleus. If they don't have a nucleus, they don't have this. They don't have chloroplasts or mitochondria or Golgi bodies. You guys are right. It's ribosome. Okay. The plasma membrane consists principally of what? Said C. Anyone else for C? That sound like a good choice? Let's do it. C is our answer. I keep trying to move this at an angle that's different. Everyone good with why that is C? Okay. Yep, alright. Huh? Yeah. Yeah, I can explain super quick. So, the plasma membrane is made of phospholipids, right? It's a phospholipid bilayer. So, first off, you know, we have to find the one that has a phospholipid bilayer. That's the only choice. And it's got proteins in it. So, yeah, it's just kind of like putting a suit and a bow tie on a very simple concept. Okay. When the concentration of solutes differs on the two sides of a membrane permeable only to water, what will happen? You think A? Yeah, maybe? Okay, well, let's think about this. So, are we talking about using energy? No, we're asking, like, what's going to happen by itself, right? So we're not going to have active transport. By plasmolysis, that doesn't make any sense here. But it, like, I like just kind of throwing words in there, and you're like, oh, I've heard that word. Water will move across by facilitated diffusion. Is that how water moves? No. And then this one says solutes and something about concentration. So, you know, let's read this because it sounds compelling. Solutes will move across the membrane from the region of higher concentration to the region of lower concentration. That sounds great. What's the only problem? Water has to move. We're assuming that solutes are not allowed to move in this case. If we said that solutes were allowed to move, that would absolutely be a valid answer. But it's not. It's A. Good job, guys. Okay. Smooth endoplasmic reticulum exhibits all the following activities except. What do we think? Oh, also we have another answer choice. I hate when this happens. You see that there? Oh no, it's too high. Okay. There we are. So we have another answer choice. What do you guys think it might be? A. So here's how my brain works on this question, okay? I'm like, okay, what does the smoothie R do? It makes lipids. And it does other stuff, question mark. Like maybe that's what my brain is saying, okay? What do I know it does not do? It doesn't make proteins because that's the job of what? Huh? Ribosomes, but on what organelle? The rough ER, right? If we're talking smooth versus rough here. So that has to be our choice. And the rest of these do happen with the smooth ER. Okay? Alrighty. Excellent. So we didn't even need that other choice anyway. Okay. Now we're talking some energy stuff. So we've already kind of gotten through chapter 7. Now we're getting into 8, just to let you know about where we're at. ATP is required for all the following processes except, okay? So ATP required, that means we're putting energy into it. It's not going to happen normally on its own. That's what we're thinking here. Active transport, do you think that that involves ATP? Yes. So we're going to eliminate that. Facilitated diffusion. No? All right. I'll hold off on that one. Microtubule movement. If we're moving things, that means that we had to put energy into it. If we're like making something actually move, then it does require a lot of energy. So we have to put energy into that. What about sodium-potassium pumps? If you hear the word pump, that also kind of indicates we're using energy. What about protein synthesis? Tricky one. Yes? Well, yeah, are we building things or are we breaking them down? We're building. As a general rule with energy, if you build something, it needs energy. If you break something down, it releases energy. That's not... Actually, it might 100% be true. But I can't validate that for sure. I know at least in this class, that's pretty much always true. So our answer is B. Good job. Again, facilitated diffusion is just any kind of passive transport that uses proteins. So like carrier proteins or channel proteins. Okay. Yeah. Good question. Yeah. Okay. All right, let's do the next one. Which of the following is an anabolic reaction? Now, before you read these answer choices, the most important thing is what? To find out what an anabolic reaction is. If you don't know what that is, you can't play here. Unless, again, you play the game of which of these is not like the others, and then you can probably start to figure it out. But let's hope that you do know what it is. What's an anabolic reaction? Think about like anabolic steroids. What do? They build muscle, right? So anabolic builds, okay? So again, we think needs energy. That's where we're going with this. We're building, we're needing energy, okay? So this one, okay? I realize that these probably look a little weird, but we're going to break this down. We have ATP plus water makes ADP and a phosphate. What have we just done? We've used ATP. This is actually the process of giving off energy from the ATP molecule. So that is a catabolic process. We're breaking something down. Okay? Now what about this next thing? Does this look familiar to anybody? Maybe? We have C6H12O6. What is that? That's glucose. Plus oxygen yields carbon dioxide and water. What do we call this? This is cell respiration in letters and numbers. Okay? Cell respiration, what was the goal? To make energy from sugar, right? We took the sugar, we broke it down, we got energy. Again, this is not anabolic. See, this is how we get tricky, right? Because rather than just coming out and saying cell respiration, I wrote the equation there and you had to remember that. Now this next one is interesting. Let's say that you have no idea what this is, okay? Because you didn't spend that much time on this. So I'm going to put that on hold, okay? Glycolysis, is that anabolic? No, that's not anabolic. Let's look at choice C. So we have carbon dioxide and water makes glucose and oxygen. What's the only thing that we know of that can make glucose? How do we make sugars? Well, we don't make sugars. Who makes sugars? Plants do. Photosynthesis. This is photosynthesis. It's the opposite of cell respiration. And this is a process that builds something. We're making a sugar. That is our answer. Okay. No, I won't keep having it sideways. Okay. All right, enzymes have all of the properties except. So now we have to think about what is an enzyme, what does it do? Do enzymes increase the rate of a reaction? Yes. Do they lower the activation energy of a reaction? Yes. Everyone's good on that. Do they exhibit specificity for certain molecules? Yes. What does that mean? So, well, yeah, it has certain R groups, but basically it's always going to work with certain reactions. That's what we mean by specificity there. Okay, so we'll get rid of that. Does it increase the amount of products that are made? Well, what did we say we can never alter with an enzyme? We always said we can't change what? Delta G. We cannot change delta G. Delta G is the overall energy of the reaction, okay? So you can't change the energy of the products or the energy of the reactants. You can't change the amount of reactants or the amount of products. None of that can be altered. So our answer has to be D. The only thing that an enzyme is permitted to do is to make what would already happen faster. That's the only role it has. Okay? Good here? Okay. What is the purpose of oxygen in aerobic respiration? What do we think? A? Anyone else for A? Anyone want to jump on that bandwagon? Well, you're going to go with A? Okay, so let's look at all the other choices and see if they're wrong, and then we'll come back to A. Oxygen is necessary to carry away CO2. Is that the purpose of oxygen being there? Not exactly, but that's a tricky one because it technically does carry away CO2, but that's not the purpose. Is oxygen used in the formation of sugar molecules? If we're doing respiration, that means we're breaking down sugar, so this is totally not where we want to be. Does the oxygen molecule just become part of the ATP? That sounds fishy as well. And does oxygen donate H+ used in the formation of NADH? No. So that's totally off base as well. So let's look at this one. Oxygen accepts electrons at the end of an electron transport chain. That sounds familiar, right? So at the end of the electron transport chain, what does it form? Yeah, it forms water. Right, yeah, so thank you for that clarification. I realized you weren't talking to me directly, but the purpose is not to form the water. This just happens. The purpose is to get rid of the electrons from the electron transport chain. You have to think about it like, you know, the electrons are moving along the transport chain, right? But there's only one spot for one electron on each protein along the way. So if we didn't have anything to pick up the electrons at the end, then there'd be a backup, and they wouldn't be able to keep moving. If they can't keep moving, protons aren't going to keep moving. If they don't keep moving, we don't use ATP synthase. You see this whole process? It's like all very cyclical. If one thing is wrong, everything is wrong. So water is formed as a result of this. That's how the oxygen takes it. Okay? Cool. All the following, we have even more energy stuff. Energy is really important here. All these produce ATP except, does glycolysis produce ATP? Yeah. Do you guys remember how many ATP? So it produces 2 net and how many total? 4. Good stuff. Okay. Krebs cycle, does that make ATP? Yes, it does. Makes a little bit, but it makes it. Fermentation. Maybe you're not sure, okay? So let's hold off on that. Oxidative phosphorylation of NADH. Or of FADH2. Yeah, because what we're saying here, again, we're dressing it up in a way that sounds different from what you've heard. But the NADH and the FADH drop off their electrons to the electron transport chain, which then leads to oxidative phosphorylation, right? So, those both make energy. Fermentation does not. Yeah. Yes, absolutely. So, anytime we have phosphorylation, I was asked to provide a definition for oxidative phosphorylation. When we have a phosphorylation event, that means that we're adding phosphate to something. So for our purposes, phosphorylation is the same thing as making ATP. Okay? So it's really the question of, in what way do we make ATP? Do we make it at the substrate level, or do we make it through an oxidation? When we take the NADH and we take an electron away, what is it called? What do you say happens to a molecule when you take electrons away from it, when it loses electrons? Well, it gets oxidized. Okay, you guys remember Leo says Gur? Maybe? Leo the lion, Gur? Lose electrons, you're oxidized. If you gain electrons, you're reduced. I'm not going to write it just for sake of time. But the point here is that when you take these electrons away from the NADH, that molecule gets oxidized. So it's an oxidative phosphorylation. There's your definition. I realize it's not like a two-word definition, but it's basically explaining how the electron transport chain works. Okay. Good questions. All right. Let's keep on moving. Chemiosmosis describes how ATP is generated from ADP. Chemiosmosis is almost completely synonymous with oxidative phosphorylation. It is the actual ATP forming event that happens with ATP synthase. It's a chemical osmosis is really what they're talking about. All of the following statements conform to the process as it occurs in mitochondria except. Okay. So we have H plus accumulating in the area between the membrane of the cristae and the outer membrane of the mitochondrion. Does that sound right? What do we call that area? Between the cristae membrane and the membrane of the mitochondrion. That's the intermembrane space. Remember that? Because this is the inner membrane, that's the outer membrane. Then we have that space in between, right? So it's kind of like mitochondria, wavy, 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 okay? So we have matrix, I'm going to just do like letters. We have cristae, we have outer membrane, okay? And then this is the intermembrane space, where all the hydrogens go, right? So that's good. That is something that happens, okay? But we have an except here, so we have to get rid of that. Do electrons travel along an electron transport chain? That's something that happens? Yes, that is something that happens. Okay, does NADH become oxidized? Yes, we just actually talked about this, so that was a good lead into this. And FADH also gets oxidized. Now, let's look at the last one, because it looks good until you find the not. See, this is the trick. I've told you guys a bunch of times, but I'm going to tell you again. There are two standard types of wrong answer choices. There is the opposite of the correct answer, and there is the same, but one word is wrong, or one piece is wrong. Okay? This is one of those. Electrons flowing through the ATP synthase channel protein provide the energy to phosphorylate ADP to ATP. That sounds great, except what? What is our issue here? Huh? Well, no, it's making ADP to ATP because we want ATP. That's what we're trying to make. Do we use electrons to go through ATP synthase? What do we use? Hydrogens. What do we also call an H plus ion? Protons. Protons. If that was the response here, then that would be a correct answer choice. You see, one word changes everything. But the rest of it sounds so convincing, you want to believe. Don't give in to the lies, the falsehood. It's not worth it. Okay. Now we're going to do some genetic-y stuff. Oh no. I realized that some of my choices are on the front and some of them are on the back. So we're going to have to just like analyze this and then flip. Okay? We have a cell. By the way, I didn't put anything from chapter 10 because, I mean, you know the amount of time we spent on chapter 10, so I figured I'd just skip that one. But it is really, really interesting when you learn more about it. Okay. If a cell has 46 chromosomes at the beginning of meiosis. All right, here we go with one of these questions, okay? We're doing meiosis here. So think about what meiosis means in your head, okay? We have 2 divisions. We want 4 cells. We want them to be unique, okay? We want them to be haploid. Think about all of these things, okay, in your mind. We can actually draw it. This was what I had recommended that you guys draw in the exam. I don't know if you did. But you draw a circle and do a quick pair of chromosomes. We have our first division. And then we do our second division. Remember this? So we're going to use this information to help us answer this question, okay? So we're at meiosis, okay? Then at metaphase 2, what would we have? Okay, so let's think about this. We've already done one set of divisions, okay? What did we separate in meiosis 1? Well, did we separate, let me give you choices. Did we separate sister chromatids or homologous chromosomes? Homologous chromosomes, right? So we have whole chromosomes, whole replicated chromosomes that went to these cells, okay? So if we're in metaphase 2, that means that we haven't gotten to our final stage yet. We're still in meiosis 2, okay? So during meiosis 2, we are at haploid and replicated, right? You see this? 2N replicated, N replicated, N unreplicated, right? 1 chromosome, 2 chromosomes. I hope we have the chromatid debate, like, killed for good. So, what we have to think here is what are we going to end up with, okay? So if we started with 46 chromosomes, how many chromosomes do we now expect? At metaphase 2. Well, we've gone from this to that. So we've gone from diploid to haploid. We now expect 23 chromosomes. And we want them to be replicated, right? Now let's look at our choices. We have 23 chromatids, 46 chromosomes. Yeah, 46 chromatids, 92 chromosomes, 92 chromatids. Now, I didn't give you an option of 23 chromosomes because that's too easy. Okay? The correct answer is here, but we have to be creative and we have to find it. Okay? Do we have 23 chromatids? No, that would mean 11 and a half replicated chromosomes. That's no good. 46 chromosomes, we already said no, we're at a haploid phase. Okay? That's no. What about 46 chromatids? Does that sound okay? Because if we have 23 replicated chromosomes, that's where we'd be. What? Why are you shaking your head? It's a good question. Yeah. But the point that I want you guys to get at is, 1, to make sure you remember this stuff, but 2, if you don't see the answer that you expect, it doesn't mean that you don't know the answer. It just means it's in a different form. So you have to be a little flexible with your understanding of the material. Okay? Now we're going to do some problems. Hooray! The ability to taste a chemical called PTC. Have you guys ever tasted that? It's like, is inherited as an autosomal dominant allele. Okay? This is very important. I don't care what the thing is. I just care if it's autosomal dominant or recessive or if it's sex linked. That's all I care about. That is my bottom line. What is the probability that children descended from parents, both heterozygous for this trait, can taste PTC? Okay? So we know that the parents are both going to be big A, little a. And if we want to know can they taste it and it's autosomal dominant, what are we looking for? Well, we're looking for big A or big A little a are all going to be valid answers, right? Because it's an autosomal dominant disorder, well, not disorder, it's an autosomal dominant trait. You're diseased if you can taste it. You know, we always talk about diseases, so you kind of just get in this thing like, oh, brown hair is like a disorder or something. Okay, so let's draw this real quick, although you may already have an idea of the results. And I apologize for my freaky A there. Okay, so what is our answer? Should be D. Everyone see how this is the case? Alright. I feel like we are making good time. We're like almost at 2 o'clock and we're like halfway through. This is good. Okay. Now we're going to deal with the most exciting organism in the entire universe, the fruit fly. So we have dumpy wings and we have normal wings. Okay, the allele for normal wings is dominant to the allele for dumpy wings. I actually gave you guys this one because it's so boring that it's like almost makes the problem harder. Because you really have to like actually pay attention to what's going on. Okay, so the normal is dominant to the dumpy wings. So we have 2 normal winged flies. Okay? Do we know their genotype? What is their genotype? How do you know? So that's what I'm saying. Yeah, so we either have big D or big D little d. Okay? Just from seeing that phrase, we don't know. Okay? We're not sure yet. So they're mated, and they produce 300 normal wings and 100 dumpy winged flies. What were the parents then? This is what we have to think about. So, I want to give us some options, okay? So, they couldn't be little d, right? That's no good. So we have to get rid of that one right away. We're going to get rid of D as well, because that one has one of them being little d. Okay? So, huh? Can't be A? Yeah, exactly. So it can't be A because we have some of them have the little d trait. So we either have this or that. So this is, I'm going to just do the first box because the second one's going to look like this. So if we have big D, and big D, little d, we get so what is the result of this cross? They're all normal wings, so is that one possible? No, what's our only possibility? C. You know what's interesting with this one is that we have a perfect 3-to-1 ratio here with our offspring, but it doesn't have to be. As long as there is one little fly with like crumpled wings, that indicates that both parents had to be big D, little d. There's no other way. Yes, even if it was like, well, we have 400, but yeah, if it was 299 normal wings, one with the shorter wings, it would still have to be this result. Even though it seems really statistically unlikely, that is the only way for it to be possible. Okay? But I made it, you know, so it kind of worked out for us. We feel good about these? I know this was kind of a rough exam for some people, but hopefully the final will not be too bad. I just want to kind of like stir up, you know, these memories again. Which of the following is true of the gametes produced by an individual with genotype big D, little d? I bring this up because you'd be surprised how many times I've actually seen people not figure out how to set up gametes. Okay? What do we think it is? You think it's B? Well, it's definitely not A. Let's go ahead and get rid of that one. That doesn't make any sense. This is kind of the one I wanted to make sure. Wait a second. That doesn't even make sense. That's the same choice as D. Yes, yes, yes. Okay, so let me just break this down very simply. Okay? What this boils down to is if you're making a gamete, that means that this is, as I had coined it before, a half a baby, right? So if we have this genotype of big D little d, then that means that when those alleles split in meiosis 1, you're going to have some gametes with the big D and some with little d. Okay? So that's how the split has to be. You can't have both of them inherited together, because then what happens is you're going to have one gamete that's going to have big D little d, another one that's going to have big D little d, and then they're going to come together to form a zygote, and it's going to be like this. And that's not good. We don't like that. Okay? So what we want here is B. I know this is a weird one, but I just want to make sure that you guys understand how to set up gametes. You want one of every allele type per gamete, is what this comes down to. Okay. So now we have what looks to be a pretty upsetting question. Let's see, we have 1, 2, 3, 4 traits. That would be a lot of boxes. If it was 3 traits, it would be 64 boxes. This should be 4 to the 4th power. I don't know what 64 times 4 is, but however many that is, and we're not going to do that. We're going to do math and figure this out. So, what is the probability that an offspring will be this? Okay? So what I'm going to do is I'm going to, rather than write out all of these boxes, which you can do, if you guys recall from my review that I did on this topic, I showed how you split up the boxes for each one. I'm not going to fill them all in, but I'm going to show you what they'll look like. So we have big A, big A, big A, big A. Okay? So what's our result here? They're all big A, big A. Okay? We have big B, little b, little b. What's our result? All big B, little b, right? Again, you can fill in these boxes, but I'm kind of just trying to cut the chase here. We have big C, and big C, little c. It should be 50-50 for either big C or big C, little c when you fill this out. And then we have big D, little d, our classic heterozygous cross. And we have that 1 to 2 to 1 ratio, right? Follow me? Okay, so let's take from the organism that we want. Okay, we want big A, big D, little d, big C, little c, big D, little d. Okay? So what's the odds of getting big A, big A? 100%, okay? What's the odds of getting big B, little b? What's the odds of getting big C, little c? 1/2. What's the odds of getting big D, little d? 1/2. Yeah, it would be, what? That box and that box. So our answer is 1/4. Yes. Sorry. Yes. No. I multiplied, yeah, I'm sorry, it's really blurry. I multiplied them all across the board. So 1 times 1 is 1, times 1/2 is 1/2, times 1/2 is 1/4. It, oh, if it was 100% brown hair and 50% blue eyes, then it'd be 50%. Yeah. Yeah, it still would. It seems kind of weird, but that would be the case. Because if you have 100% odds of getting something, then that means you don't even have to consider the possibility of getting it because you will get it. That's kind of how you have to think about it. Yes? Would I not use what? You could absolutely use the 2 to the n rule here. Well, I don't want to do the whole thing right now with the 2 to the n, but we can look at how many gametes they would make. Because this one, let's see, we have 1 heterozygous pair actually. So 2 to the 1 would be 2. And this one, we have 2 of them, so it would be 2 to the 2, which is 4. So you'd have a 2 by 4 box, but it would still be a lot of writing because you'd have a lot of letters. But you can absolutely do it that way, and I can show you if you still want to see it after the review. Okay? Yeah, that's another way of doing it. I just wanted to kind of show you guys this way because it seems to be a little bit quicker sometimes. Yeah, either way will absolutely work. Okay. Now we have what appears to be a very long question, but it's actually not so bad. In domestic cats, 2 alleles of a sex-linked gene code for hair color. What does this sound very similar to? 1 allele codes for orange hair and the other codes for black hair. Cats can be all orange or all black, or they can be, oh look, it's calico. Ta-da! So I basically described the calico phenomenon in this question. A coat characterized by randomly arranged patches of orange and black hair. Okay, with respect to this gene, all of the following are true except what? Okay? So now we have various crosses that they want to experiment with. What we're going to do, because they're not asking for probabilities, they're saying, can this happen? Okay? So we're going to just very quickly look and see if they can happen. So we have a black female. What will her genotype be? So remember we have to do X's. XB, XB. Okay? And then the male will be orange. So what's his genotype? XOY. Okay? Can they produce a black male cat? Well, if it's male, he gets the Y from his father, right? And he gets the X from his mother. So can we produce a black male cat? Yes, we can. This is how we do this. A black female, okay, we'll do the same thing. And an orange male. Can they produce a female calico? Well, what does it take to be a calico? 1 black, 1 orange. We can pull 1 from each parent. So yes, we can make a calico. And you can actually do it as a cross, but I'm just kind of showing you guys the possibilities here. Alright, that is true, that is possible. Have I lost anyone so far? Okay. We're going to keep moving. Okay, a calico female. So, you guys can see what I'm writing over here. Calico female is going to be XBXO. And then the black male is XBY. Okay? Can they produce a calico? A female calico? Yes. We have O, we have B, right? So it's the same kind of deal. I'm going to start doing these below because I'm kind of running out of space here. A calico female, XBXO, and an orange male. It all kind of starts to feel very repetitive once you understand what's going on. Can they produce a female calico? We have an O, we have a B. Yes. Yes, they can. So now we get to our last choice, which we're hoping is wrong, otherwise we have an issue. A calico female, sorry, let me do B first just so we don't get confused. XO, and an orange male, again, can produce a male calico. Huh? Yeah, so this is actually kind of a trick question here, because if you would scan through the answer choices, you would see that in normal circumstances, now I'm not talking, we do have exceptions here, but in a normal circumstance, we cannot have a male calico, okay? Because this individual would have to be XB, XOY. What do we call this? This is Klinefelter syndrome. But we're assuming normal circumstances, so that's going to be our not here. But you can actually do this and have, other options as your not, yeah? Yeah. Yeah, I mean, that definitely is a possibility there, and you can do that, yeah. Okay. Any questions here? Okay. Let's keep moving. This is a really interesting question that I like to have you guys think about. From which parent or parents did a male with red-green color blindness inherit the defective allele? That's a weird question. Color blindness, how is this inherited? It's sex-linked, right? Okay? So what is the genotype for a male with color blindness? X, I guess we can do C or whatever. X little cy, right? So, where did he get the defective allele from? Had to be his mother. Because, where did he get the Y from? His dad, right? So there's only one option. So, and this always gets a laugh, but if you are a male and you're colorblind, it's your mom's fault. I mean, not really. I don't think it was her choice directly, but I can say it about this because color blindness isn't like a super severe thing. But if it was anything else, it'd be kind of bad. But yeah, so it actually, because he only gets the X from his mother, then you can trace it back so you know that she had to be at least a carrier. Okay, now we're getting into, so we have one more question related to genetics. This is a concept question. A human genetic defect that is caused by non-disjunction of the sex chromosomes. What do we think? You'd think that. However, Down syndrome is non-disjunction of what chromosome? It is trisomy 21, which is a body chromosome. Yes, we can. So non-disjunction is a failure of chromosomes to separate. So this is a very weird word. Okay, junction means coming together. Disjunction means coming apart. Non-disjunction means they don't come apart. So that's sort of a weird way to think about things. But Down syndrome results, it's basically, let me redraw our situation here. Okay? So if we have our chromosomes in meiosis, alright, let's say, how do I want to do this? Let's say that they separate normally, okay? Actually, I'm trying to decide what I want to do here. Okay. Let's say that they separate normally in meiosis 1. And then in meiosis 2, bless you, these ones separate normally. And then these ones don't separate at all. And we have one cell that has a pair of replicated chromosomes and one cell that got the short stick here. Okay? So what the result is, okay, think about this. you're either going to have, because these are all gametes, right? This is not the future organism, all right? We're going to put these together with another, hopefully normal gamete. We always assume that they're pairing with a normal gamete. The odds of having two non-disjunction events in both parents and those gametes meeting each other is like winning the lottery squared or something. Totally legit statistics. But anyway, it's the idea that if this gamete goes and pairs with another, then this individual will be lacking a chromosome somewhere, whatever one didn't separate, okay? This individual will have one extra chromosome, okay? So Down syndrome would be that situation. We also have Turner syndrome, which is when an individual is XO, and the O just means that there's nothing there. So they only got one X chromosome. This is a monosomy event. I'm going to mention that. So we have trisomy and monosomy. You do not want either of those. You want two. Disomy? I don't really know actually what that would be, because it's not something they talk about. Huh? Yeah, it would be normal. So our answer actually is going to be D, because that is the sex chromosomes. Alright, let's look at some of these. Sickle cell is not related to any of this. Sickle cell has to do with a missense mutation, right? We get one different amino acid. Hemophilia is sex-linked, and color blindness is also sex-linked. So I threw in some things that like sounded vaguely familiar but were unrelated. So I like doing that. And then Down syndrome was really the one to make you think, because you're like, ooh, non-disjunction, Down syndrome. But you have to be very careful. So I appreciate that you said that, because that's, you know, a good thing to think about. Everyone good with this? That's also how we get Klinefelter males. Because if this was the X's, then if that paired with a Y, then we have two X's and a Y, right? Okay, and now we're going to do DNA. So just like that, we're done with genetics. Okay. The two strands of a DNA molecule. Also, by the way, we have crossed the 1 hour mark. I realize that, like, probably starting to feel a little restless, but I'm going to ask that you hold on just a little bit longer. I'm going to try to finish early, and then we can do some extra problems if you want. But we gotta hold tight. All right, the two strands of a DNA molecule. Let me draw a little DNA molecule for us. So it's not going to be twisted because I can't really do that. But it's going to be straightened out, which is not something that happens in the real world. But it looks like a ladder, right? But it's actually like, boop. So we have this thing. We have the 5 and 3 and all that. We want to know the two strands. We're talking about each side. What are they connected by? What is in the middle? You saying hydrogen bonds? Well, let's take a look. Is it hydrogen bonds between codons and anticodons? That sounds a little fishy, right? Where do we have codons and anticodons? Yeah, so specifically in translation is when we're talking about these. Okay? Do we have hydrogen bonds between the bases of one strand and the bases of another? Yes? You guys are pretty confident with that? Let's see if you're right. Hydrogen bonds between deoxyribose sugar of one strand and deoxyribose molecules of a second strand. Well, where do we have the sugar? The sugar and the phosphate is in the backbone. Right? Sugar, phosphate, sugar, phosphate. But in the center is where we have the bases that are connected together. So that's not good. What about covalent bonds between phosphate groups? There's a couple issues here. For one, the phosphates it's like phosphate, sugar, but also we're not talking about the outside, we're talking about the inside. And then covalent bonds between the nitrogen bases. If that was the case, because you guys remember covalent bonds are stronger than hydrogen bonds, right? So if we had covalent bonds down the center of this, we would never be able to open up DNA to make a new strand of DNA. That wouldn't happen. So that one's not good either, and yes, you guys are right. Yeah, so the bonds here, they are a type of covalent bond. They're called phosphodiester bonds. But, yeah, so you don't really need to worry too much about that, but that is what they're called. Okay, all of the following combinations of nucleotides are examples of normal base pairing except for what? Ooh, you're right on this. Well, let's see. Adenine DNA with thymine DNA. Does that sound okay? Yes. Guanine DNA with cytosine DNA. Does that sound good? C and G, yeah? Thymine RNA with adenine DNA. What's the issue? There's no thymine in RNA, so immediately we have a hiccup. And I think by just hearing it out loud, it may be a little bit clearer, you know? It's like thymine and RNA. But let's look at these other ones, because E does sound weird, so let's look at that. Cytosine and RNA and guanine and DNA. Is this possible? Yeah, because when we're making the RNA, we do have DNA and RNA that's connecting with each other. So that's okay, we're making a transcript. And then same thing here, uracil is going to A. A and U go together, right? So that's fine. So really, bless you, our issue here is with C. Huh? What? Oh, cold, yeah. It's so cold in here. Oh. You know, it's actually really interesting because the majority of the time when people do the survey for study union, instead of talking about the session, they're like, room was cold, sad face, and then they give bad ratings for the whole thing. It's like, well, you know. Okay, all of the following enzymes are involved in DNA replication except, this should be fresh on your brain. Well, we have helicase, splits open the DNA. We have ligase, seals the Okazaki fragments. We have polymerase 3, it's going to build our DNA. We have RNA polymerase. That is transcription. Would you look at that? And we do have primase. I want you guys to be very careful. Not that I think you're specifically going to get something insanely tricky, but even in classes to come, if you have this topic again, which if you're biomed or anything like that, you will. Primase and RNA polymerase sound very similar by definition. So you just want to make sure that you can really pick out the nuance difference between them, because it won't always say this one's in replication, this one's in transcription. So just, for your information. Okay, what do we get from translation? What do you feel like you want to say? Everyone? A lot of people want to say amino acids. But are you making amino acids? No. In translation, what we do is we take already made amino acids and we're going to put them together in a very specific order. And when we make this order of amino acids, what do we call that molecule? We call it a polypeptide. Okay? All right. Oh, by the way, normal rules will apply with this today. If you guys want to get pictures at the end, you are more than welcome to. Wait, is this... Oh, I haven't done the back. Okay, never mind. All right, so this one we're going to have to flip over. An mRNA actively being translated in the cytoplasm would have all of the following except what? Would it have a poly-A tail? Yes. Would it have a 5' cap? This is basically our run-through of processing. Would it have exons? Would it have introns? Not so much. Would it have RNA nucleotides? Yes. It's made of, that's what the exons are made of. I'm very enthusiastic. Okay. Questions there? Anything? You know that it's gone through processing because it says it's being actively translated. If it says it's being translated, that implies that the stages have been done correctly previously. Because we can't, I mean, there's a whole crazy series of assumptions we could make. We could be like, well, what if it didn't happen right? Or what if this happened? Or what if that happened? But we're like assuming that things went well, you know? That is a good question, though. Thank you for asking that. Okay, both viruses and prokaryotes contain, they both contain nucleic acids. A virus has what components? Yeah, a virus is this. It is some kind of nucleic acid and a capsid. Does it have ribosomes? Does it have a plasma membrane? No. Viruses have a protein coat, but do prokaryotes? No, they have a plasma membrane. You see how we don't have that crossover there? And then what about a peptidoglycan wall? That's prokaryote only. So our answer is A. Great job. We're just doing a basic run through of like compare and contrast. Okay? Alright. Which of the following is possible for the genetic material of a virus? You think B? Any other thoughts? You know, our answer is actually E. And this is weird. D is an interesting choice because it is the same as B. So this wouldn't really be a fair test question anyway. But what I wanted to get at with this, because I feel like not a lot of people realize this, is that a virus can have either DNA or RNA, and that DNA or RNA can be either single or double-stranded. The reason that we learn about viruses at the end is because they break like every single rule we've ever learned about anything. They do, exactly, they do whatever they want, and they are not, according to current scientific majority opinions, they are not considered to be alive. And there's a lot of reasons for that. So they kind of just have their run of the microbial world. They can do whatever they want, which is sort of frightening. They're a lot more difficult to deal with than bacteria, incidentally, like from a human fighting perspective. But yeah, so they can have any of those options. They can also, I didn't put single-stranded RNA, but they can have that as well. So like I said, there's 4 options. There's actually one virus, it's called Meiosis. I only found this out like in my upper level classes. It has DNA and RNA. So, you know, there's that as well. I don't know, I don't remember. You should look it up though. But yeah, anyway, let's keep going. Now we're gonna do blood stuff. But not really, actually. So this is interesting. It's actually more of like an evolution genetics kind of question. The B blood type allele probably originated in Asia and subsequently spread to Europe and other regions of the world. This is an example of what? Well, let's look at these options, okay? So we're going to define each of these. Artificial selection, do you guys know what that is? Yeah? So it's like natural selection, but not done by nature. It's primarily with humans, I'd say. The best example of this would be dog breeding. Okay? All dogs are descended from wolves, right? I have a Yorkie. He's like 8 pounds. But he is descended from wolves, and that's kind of weird for me to think about with my little yappy dog. But the reason, or the way that Yorkshire Terriers came to be is because there were people who were like, hey, we want super tiny dogs. So they would pick specifically the dogs, and I mean, of course, this has happened over many, many, many generations, but they would pick dogs that were like the smallest of their litter, and then have them breed, and then they'd pick the smallest of that litter, and they would keep going from there. Or like any other traits, if they wanted softer fur, if they, whatever it is. But that's probably one of the best ways to think about it, because it's not that they can survive better in the wild. My dog like freaks out if there's stairs, so like he's not equipped for the wild. However, there's plenty of Yorkies. Why? Artificial selection. Okay, so that's my example. Natural selection. Now we have Darwin. Okay, it's the idea that it's a very similar thing where there's differences in the world, right? So we're all different, right? You look around, we have different characteristics, different animals have different characteristics, and so maybe some of those characteristics lend you to be better able to survive than other people's characteristics. Maybe you can run a little bit faster, so if there's like a tiger chasing you, then you might run faster than your buddy who's going to get eaten by a tiger. You survive. So I don't really know why I'm doing this. So the people, back in, time, the people who were able to run faster, like maybe they were able to survive better, and so then now people are faster than they were. That's just like, a thought. One of the key things though with being like evolutionarily fit according to Darwin is not just survival, but it is reproduction. You have to survive long enough to have babies. Otherwise, it's no good. It's the idea that someone is more evolutionarily fit if they live to be 25 and have 6 kids than if someone lives to be 99 and has no kids. You have to pass on your genes. That's like, the purpose of life instinctively or something. Maybe not. But that's the idea anyway. Okay, you guys get what I'm getting at? So that's the idea with that, okay? But we're not talking about that, so we're down to our other choices. Genetic drift is actually random fluctuations in genes that just happen, okay? So I'm going to just put random because that's the most important thing here. So that's not going to be effective for this choice because we're talking about people traveling across the world. So that's something that's deliberate. That's actually gene flow. Is when and there's probably a better definition of it, but the idea is that the distribution of genes for an organism changes based on migration. So, if I like to use like us as a microcosm as an example. So, we consider the prospects of there's like an alien invasion happening right now, which is really a bummer because this would be like the last thing you'd be doing potentially. And so, we're able to survive, but we can never leave this room. And somehow we have like food and stuff, right? So, the genes that are in this room would be the only genes ever for this population. But then, oh look, they defeated the aliens. Yay, we get to leave. So now you go and blend in with the rest of the world. Okay, maybe some generations later. We've been here for like 40 years. So that's the idea, gene flow. Okay? And then sexual reproduction. Pretty sure you know what that is. But that is going to provide variety in offspring, and we've learned all those reasons why. Okay? The appearance of a new mutation is what? Well, this is the interesting thing, is we kind of have the cause and effect mixed. So natural selection happens as a result of mutations. Because there's mutations that give us different characteristics, and then those different characteristics render us differentially able to survive, right? So we kind of have that a little bit backwards. Again, artificial selection doesn't really come in here. If sexual reproduction created new mutations, that would probably not work so well for animals that reproduce that way. And then usually a beneficial event. Unfortunately, I feel like all of the craze of superhero movies has taught us that you get exposed to gamma radiation, you're going to get like cool new powers or something. But normally mutations are bad. There's a very rare occasion where mutations are good, and then it's like, oh, we have a new trait. But they are completely random. And that's what we're getting at here. Now, you can have increased or decreased likelihood to get mutations depending on what you're exposed to and your life and your genetic predispositions and whatever, but the actual mutation event is random. It can happen to anyone. Your ability to fix it can depend on you, so there's options there. So that's what we're getting at. Okay. And there's a lot more of those definition things, but I didn't really feel like we would have time to get to them, and now we can spend the rest of our time doing Hardy-Weinberg. So, good stuff. All right. So now we have sheep, which are cool. I like sheep. We have 9% white sheep and 91% black sheep. Now I realize, okay, never mind. I was going to say, I didn't say if it was dominant or recessive, but I did. What is the frequency, let me not get ahead of myself, what is the frequency of the black wool allele if the black wool allele is dominant and the white wool allele is recessive? Okay? So let's break this down. Alright? What do we want to find? What are we looking for? What did I say we're trying to find? Frequency of the black wool allele. Okay? I'm going to take this space up here, and I'm going to write something here. P plus Q equals 1. Yeah, you can see that? No. That's not what I intended. P squared plus 2PQ plus Q squared equals 1. Please make sure that you know these by the time you take your exam. And then let's write what they represent. So these are allele frequencies. Okay? P represents what? The dominant allele. So I'm going to just do A, because I like A's. And then Q is what? Recessive. It's the little a. Okay? What does P squared represent? What does P squared represent, guys? Homozygous dominant. This one is heterozygous. This one's homozygous recessive. Okay? These are genotype frequencies. Here's the biggest hint that I can possibly offer you for Hardy-Weinberg. If they are talking about individuals or people, okay? If they're talking about actual living creatures, then they're talking about one of these, right? Because you have to have a genotype. If they're asking about alleles, then they're talking about one of these, okay? So if they talk about the frequency of blah, blah, blah individuals, then you can't just have Q, because Q is not an individual, okay? So that's really going to affect our numbers here. Okay? All right. Did you guys have a question over there? Or you good? Yeah? Well, why don't we do some of this, and then I'll see if you still have that question. Okay? So let's go ahead and try and solve this. So what letter do we want here? If they want the frequency of the black wool allele. We want the dominant allele. We want P. All we're asking is, what is P? That is what the question is asking. Does everyone understand what the question is asking? That is your first step. That is really your only step. Because if you don't know that, then you can't go anywhere with this, okay? So, we have, let's see. We have 0.91. I'm taking this out of a percentage, putting it back into a decimal here. 0.91 is going to be black, okay? What is the genotype for black? So I'm going to do A's again. So we have big A, but is that the only choice? No. Or little a, because it's a dominant phenotype, not a dominant genotype. You cannot guarantee what the phenotypes are. Huh? 91% of them are black, and the black allele is dominant. Oh, yeah. That's what I meant. You guys know that. I'm sorry. Yes. No, it's okay. Yeah. Yeah. Okay. So that is these individuals. Yeah, sorry I put little a, little a. I don't really know what I'm doing. I do know what I'm doing, but I don't know what I'm writing, I guess is a better way to say that. Okay, so then 9% is white. So that's 0.09. What genotype is that going to be? Got ahead of myself. That's our little a. Okay? Now, because this one is uncertain, we can't use this information. Do you guys understand that? We can't use the information that we don't have a guarantee on. But we have a guarantee on this. And what is little a represented by? Q squared. Why isn't it just Q? Because this is actual, well, they're sheep, but this is actual sheep. This is actual living creatures. Okay? So you have to do Q squared here. So then we want to find Q, which is going to be the square root of 0.09. What is the square root of 0.09? 0.3. It's not 0.03. Please make sure that you guys are very careful with your square roots and stuff. And I don't mean that as an insulting thing. If it wasn't for how often I do Hardy-Weinberg, I wouldn't know these numbers either. So if Q is 0.3, is that our answer? No, we want P. P is 0.7 because P plus Q equals 1. So yeah, that's what we would do with this equation. The most important thing from like these equations up here is to realize that when you find P and Q, it's not about plugging it into the whole thing. If you plug every single value into here, what are you going to get? Guys, let me get your attention. So, if I plug in P and Q in here, what will I get if I solve that whole thing out? It'll equal 1. So if you do that, that's not going to help you. You need to just extract like the information that you need directly. And then you want to find either P or Q, and then you're good from there. You know what I mean? Okay. Any other questions with this one? Yeah. Why I didn't use P squared? Well, let me put it to you this way. The 0.91 would equal P squared plus 2PQ. Do you want to try and solve that? You see what I'm saying? It's like a rhetorical question. This is why we don't use the P value or the dominant value, because then we end up with this grossness, and then you have two variables, and that's something we don't want to deal with. Yeah, so the P plus Q equals 1 you can use as long as you have either P or Q. We didn't have P or Q. We needed to find one of them, which we did, and then we found the other. Yeah. So you kind of have to play with these a little bit, but that's the really important thing, is to realize that you can't just set the 0.91 equal to P squared, because that's not a fair representation. There is some Q there, and that's the problem. Okay? So, let's do this one. In a population of 1,000 superheroes, 360 have telekinesis, which is recessive. Assuming Hardy-Weinberg equilibrium, how many individuals are heterozygous for flying, which is the dominant trait? And then we have how many would be heterozygous if telekinesis was dominant. Okay, so let's start with our first question and not get ahead of ourselves. Okay? 360 have telekinesis, which is recessive, okay? These are individuals, so what is their genotype? They're little a, or whatever. So what are we going to represent that as for the equation? Q squared. Everyone good with the Q squared again? Because these people that we say have the recessive trait, they can't just be Q, because that's saying that your genotype is little a. That's saying that you don't have two parents. You see? You have to have two alleles, so you have to have one of those. That's where this comes in. Okay, so we have 360 out of 1,000. What does that equal? 0.36. Take square root, Q equals 0.6, right? Okay, how many individuals are heterozygous for flying? So how do we find heterozygous individuals? 2pq, right? So it's 2 times 0.4. This right here, our p is going to be 0.4. And you can do this in more steps. I'm just kind of abbreviating it slightly. So we have 0.4 times 0.6. Yeah? Can you not see? What? We do the middle step? Okay. Q is 0.6. Okay. So 0.6, 0.6 plus p equals 1. Solve for p is what we did. I plugged it into the first equation. Yeah. I'm sorry. I kind of jumped the gun there. Okay. Everyone good with how we got p? Alright. So we plug that in, and then what do we get? 6 times 4 is 24, times 2 is 48. 0.48. Is that our answer? Can you have 0.48 individuals? It's like a really graphic murder scene, and that's also not representative of a population, right? So we have this out of 1,000, so how do we represent that? We have to multiply it by 1,000, which gives us 480. You see? So we have 480. So it's pretty clear what our answer has to be, but let's do the other one. All right? So now we're saying how many would be heterozygous if telekinesis was dominant? Okay? So now we've changed the game. Because our 360 out of 1,000 now equals what? It's the dominant, right? So that would be p squared plus 2pq. Do you want to solve that? Do you want to solve that? I don't want to solve that. Let's not do that. Let's instead look at the other individuals. If 360 have telekinesis, how many is the other group? 640, and that would be q squared. So q squared is going to equal 640 of 1,000. You see how we flipped this situation, and now it's different? Yeah? What aspect? Yes. So, we said now, because at first we said 360 were recessive, which made them q squared. Now we're saying, this is a new question, but we put it in the same one. How many would be heterozygous if telekinesis were dominant? So now this is the dominant trait. The 360 are all dominant individuals. So that means that the other individuals have to be recessive now. So that makes them, the 640, that makes them q squared. And now we have the exact same process, so we'll do this quick. So q is going to equal what? Q is 0.8. What's p? 0.2. 2pq equals 0.2 times 0.8. Oh no, I forgot my 2. I'll put it at the end. 2 equals 0.32, right? 0.36? Wait, E times 2 is 16. Ah, okay. 0.32. Okay. Then, what do we have to do? We have to account for the 1,000. So our answer is B. I know that went super crazy quick. But does this kind of feel okay? You're not going to get a question that's that many layers, but I'm trying to throw it all at you at the same time. And then when you just are asked about one piece, you'll be like, yes, I can do this. So know this, and then you can definitely do that. You know what I'm saying? All right, let's try a couple more. We can go pretty much right up to the wire. I thought we'd have time left at the end, but it looks like we're not going to. We have about 10 minutes left. And we have 3 questions, so we'll try and get through them. Now we're on Mars, which is exciting. Green skin color is dominant to purple. We have 1,000 Martians. 200 are homozygous dominant and 100 are purple. So, we want the frequencies of the dominant and recessive allele. Now this is a very interesting one. I'm going to actually draw out what we have, okay? Homozygous dominant, bless you, is 200. Agreed? That's what we said. 200 are homozygous dominant. Okay, for green. We said green is dominant to purple. So if they are purple, what does that mean their genotype is? Recessive. Little a equals 100. Now we have this interesting situation where we have the total population. Can we figure out how many heterozygotes there are? Well, we have 1,000 total. We take out the 200 for homozygous dominant, and we take out the 100 for homozygous recessive. What are we left with? 700 heterozygotes, right? I'm not doing any kind of wizardry here. It's all with the information that we have. So, we now have this exciting situation where we actually have every genotype. So at this point, in order to find the frequency of the dominant and the recessive allele, it's more accurate to not use Hardy-Weinberg, but instead do the splitting alleles technique. And that was what she had talked about in class. Remember that? The key thing for you guys to pick up on your exam to see if you have this type of question is, do you have every genotype present? If there is nothing for you to solve for, then you're going to have to do splitting alleles. Okay? Because in that last question, I'm going to just bring it back real quick. In this question, this one with the superheroes, did I tell you how many heterozygotes there were? I didn't tell you how many of that dominant population were homozygous or heterozygous. That's what we're basically trying to figure out with Hardy-Weinberg. So if you already have that information, then you don't have to solve for it now. We now are able to actually accurately depict what's happening. Okay? And there's 2 ways to do this. I will show you both ways. Okay? Whichever one you like, just stick to it and ignore the other. Okay? There's way number 1, which says that if we are looking for big A, okay, then from the first group, we're going to pull all of them, right? Because all of them have big A, right? That's everything that they're contributing. And then from the little a, they're not going to contribute any big A's, right? No big A's. And then from this group, they're going to contribute big A's, but only half of what they're contributing is a big A. So we're going to put 350. Okay? So we get 550, and then we take it out of our total population, which is 1,000, which gets us 0.55. Okay? I'm going to show you a different way now to get the same thing. This idea says that from the homozygous dominant individual, they're going to each contribute 2 dominant alleles. So we count every allele. So we'll count 400 alleles here. We still get none from the homozygous recessive, and then we're going to get 700 dominant alleles from these guys, because each of them contribute 2 alleles. Okay, so that's going to get us 1100, and then we take it out of the total number of alleles. If every person gives 2 alleles, how many total alleles do we have? 2,000, which still gets you 0.55. I like this one because the math is easier, but it's up to you, whatever sticks with you. So then what does our answer have to be? Our answer is A, because you can find the recessive either by doing the same process or doing P plus Q. You can actually, yeah, it might just be better for me to say, go ahead and do the full process out, because then you're going to be like, well, that's Hardy-Weinberg, and I don't want to get into why that is possible, so yeah, that will be our answer. Is this okay? Again, either way will get you the right answer, so just stick to whatever works for you, whatever makes sense in your head. Okay? And now we're going to finish up with actually a couple last-minute questions here. So this one says, because of human predation, the sizes of and genetic variation in populations of most whale species is declining. Okay? What is this an example of? You guys have thoughts? This is going to be bottleneck. So it's kind of one of these things that you have to just learn, but both bottleneck effect and founder effect result from having a very small population. So this is, bottleneck effect is the idea, again, I'll use this room, okay? So it's the idea that a meteor strikes this building. I'm like very cataclysmic in this room right now. A meteor strikes this building. Everyone dies, except for you guys. This table right here in the front. You guys survive. Yay. Okay, so you're like the last of our study union species, okay? So, as a result, the distribution of genes that you guys have is probably not representative of everyone else's. Like, what if there was one person out here who had, like, some really cool eye color, like some kind of bluish, grayish eye color, right? And none of you guys had the gene for it, but now they're dead. So now that gene is gone. You know what I mean? So, that's how that happens. Founder effect is kind of like a voluntary thing. So it's like a couple members out of the species go out and decide, hey, we're going to go off on our own and start a new society. And then there's also not that many genes. Okay? But because we're talking about the species are declining and they're being reduced and they don't have as much genetic variation, that's how we get there. Okay? And then the last one. A recently introduced species of seed-eating birds occupies an island where small and large seeds are available. Beak size in the bird population varies from small to large, allowing some birds to be more successful at eating small seeds. So some are more successful with small seeds. Others are more successful at eating large seeds. And birds with the in-between beak size, again, get the short end of the stick here, they have to exert additional effort to eat seeds. So what kind of selection is this? This would be called disruptive selection, because it's selecting for the two extremes rather than the normal. So like a normal bell curve in a college class is a stabilizing selection. So that would be the idea that they're going to normalize the grades until most people have C's, okay? And a directional selection goes to one extreme or the other, okay? We are going to go ahead and wrap it up here again. Remember, we do have 2 SI sessions tomorrow. Please do either scan the code or write that link down and take the survey for us. If you have the opportunity, I'd really appreciate it. If I don't see you guys, thank you so much for a wonderful semester, and good luck with everything.